Hello everyone, welcome to my channel. So today we'll be discussing a little bit about a process called glycolysis. Now glycolysis is a very basic biochemical pathway, and it is a central pathway for many of the biochemical things. It produces lots of energy, and also it produces a lot of intermediates which are important for production of several other products that are important for our body. So today we'll be just discussing some of the overview, and we will not go into the details of anything, but we'll just see the overview of what is glycolysis and little introduction. So first is glucose as the fuel. So what do I mean by fuel? Fuel is anything from which you can extract a lot of energy, like petrol is a fuel, diesel is a fuel, like that. For living organisms, glucose is the fuel. So we know glucose is a 6 carbon molecule. It is a sugar molecule with 6 carbons. There are a lot of other sugar molecules with 6 carbons, but they are not the central pathway. It is the glucose metabolism that occupies a central position, and all the other sugar molecules somehow feed into the glucose um metabolism, and they get metabolized in the same steps as glucose. So they have to feed into the metabolism. But glucose, this is occupies the central position. Now, like any other fuel, like petrol and diesel, the glucose has a lot of potential energy, and it is rich in potential energy. So if you can tap this energy, you can convert this energy into some other form of usable energy. So in body, what happens is that we split and oxidize the glucose, and we convert it into a form of energy called as ATP. ATP is the energy currency, so you can use it for different energy requiring processes in our body. So the complete oxidation of glucose yields about 2800 kilojoules per mole. So 1 calorie is about 4 kilojoules per mole. So you can calculate how much that is. Now, before going into proper pathways, let us know an overview of metabolism in our body. So our body needs energy, and it needs energy all the time for growth and all this other different processes that it has to do. So it needs energy continuously throughout the time it is going to be alive. So this energy is we are obtaining mostly from our diet. So our diet contains carbohydrates, proteins, and fats. Those are the major providers of energy. So carbohydrates, when they are digested and assimilated in our body, we get a lot of simple sugars. Example: glucose, fructose, galactose, mannose, and all we get. But majority, the most important and most abundant is the glucose. Then from proteins, all the proteins are converted into amino acids, and all the fats are converted into fatty acids and glycerols. As you can see here, they can be converted into fatty acids and glycerols. Now all three, all the glucose, amino acids, and fatty acids, they are all converted finally into an acetyl CoA. Okay, and this acetyl CoA gets oxidized in a series of reactions that is called as citric acid cycle, or tricarboxylic acid cycle, or even Krebs cycle, which you might have heard. Now this part is called as final common pathway. That means all the carbohydrates, proteins, metabolism, they converge here, and it is from here we get all our energy. Now the glycolysis, where does it fit is in this process conversion of simple sugars like glucose into acetyl CoA. So glycolysis as such, the end product is pyruvate, and pyruvate has to be again oxidatively decarboxylated to produce acetyl CoA, and we'll be seeing these reactions in detail later in the slide. But the overview is this, that all of these gets oxidized and they are all converted into acetyl CoA, and acetyl CoA gets further oxidized in tricarboxylic acid cycle, and it produces ATP or reducing equivalents. This reducing equivalents have to go through the electron transport chain, and they also will produce ATP at the end, and that process is called as oxidative phosphorylation. So here is where we have our glycolysis. As I told you, see, this glycolysis is very important because this is the central pathway. You can see that it is present in the center, and it is related to pentose phosphate pathway. It is related to tricarboxylic acid cycle. This is a tricarboxylic acid cycle, and you can see it is also related to triacylglycerol and glycerol through this. So there is this is the central pathway, and the understanding of this pathway is essential to understand further what happens to our metabolism. So uh, earlier we saw that for our energy needs we are depending on the dietary food, but we are also depending on our internal storage. So we also have plenty of internal storage, and in our body the storage of glucose is called as glycogen, and in plants it is called as starch. So these are nothing but high molecular weight polymers of glucose. So in needs of emergency, in times of starvation, times of emergency, in times of fasting, you can also derive glucose from these, not only from diet but from our internal storage also. You can get these energy giving products. So versatile precursor, what we mean by that is that glucose is not just a fuel. It is not only used for producing energy, but as we have seen in the previous slide, it is a central in our metabolic pathways. So it also produces so many intermediates, which are in turn precursors of many of the essential components of our body. So huge array of biological intermediates are formed. Like you can see here, glucose forms the carbon skeleton of amino acids. It forms several parts of the nucleotides. It forms pyruvate. It forms storage forms like glycogen and this thing. It forms extracellular matrix and very very important. And it also forms fatty acids. So it is a versatile precursor also. So glycolysis is not just energy yielding process, but it also produces so many products which are precursors of many other important pathways. So there are so many pathways emerging from these. And in evolutionary perspective also, glycolysis plays a very important role, because you can understand that as we evolve from a single cell to a multicellular species, um, uh, during the evolution of this smaller species, these bacteria, the earth did not have this much of oxygen. Right now we have about 20% of oxygen is there, but earlier we didn't have this much oxygen. So at that time, whichever animals evolved at that time, they had to rely on a process that will yield energy without the presence of oxygen. And that process is called as anaerobic glycolysis. Anaerobic means without air, but technically it means without oxygen, not air. So air was always there, but oxygen amount was less. But still we are calling it as anaerobic glycolysis. And this process is also called as fermentation. And this process is found in single cell all the way to the highest evolved form, like humans. So the core pathways of this process are retained even in the higher animals, and most of the reaction sequence is also conserved. The sequence is also conserved, and also the structure of enzymes are also conserved, you can see. And there is difference is there, and the difference is in certain pathways and also mainly in the regulatory aspects. So in the lower animals, this amount of metabolism is itself was enough, but in higher animals we have so many other needs other than energy. And even in energy we have so many much more higher needs of energy than a single celled organism, right? So what happens is that the regulatory aspects changed. So we have made ourselves more complex, and our regulation is also more complex. Um, so that is the evolutionary aspects. Now let us go to glycolysis proper. So what do you mean by glycolysis? Now the word glycol is derived from the Greek word glycus, which simply means sweet, and lysis means splitting, which we already know. So glycolysis, what do you mean by glycolysis? The definition can be given like this: a single molecule of glucose is degraded, or in other words oxidized, in a series of reactions to yield two molecules of 3-carbon compound pyruvate, and along with that a few ATP molecules and a few NADH molecules are produced. That means it is not just production of 3-carbon compound pyruvate, but the end product is also several ATPs and NADH. That is the major role of glycolysis. Now, some of the free energy released from glucose is conserved in the form of ATP and NADH. So throughout this process, what we are going to see is that this glucose is a fuel. It contains a lot of potential energy between its bonds, and we are manipulating these bonds. We are rearranging them and manipulating them in order to create ATP and NADH. So that is the basic funda. So where does this happen? That means where does it take place? So it takes place in the cytosol, that means cytoplasm, and it takes place in all the cells. As I mentioned before, all the cells of our body utilize glycolysis for energy. Now, there are certain cells in which the only energy giving process is glycolysis, especially cells like your red blood cells or erythrocytes. They do not have mitochondria. That means they cannot undergo oxidative phosphorylation. So, the only way in which they could gain energy is by using a form of glycolysis called as anaerobic glycolysis. And this is the only process that could happen with or without oxygen. All the other oxidative pathways will cease to exist if there is no oxygen. But glycolysis can keep on going even without the presence of oxygen. And there is about 10 steps in the glycolysis, and these 10 steps are generally divided into two phases. Some books gives 3 phases also, but generally they are divided into 2 phases, 5 steps in each of the phases. So we have come to the glycolysis uh, steps. So first is a preparatory phase. So it consists of the first 5 steps. You can see the first 5 steps. So here basically what happens is that glucose is split into a 3-carbon compound, and it is first phosphorylated and then split into 3-carbon compound. This is the total funda: glucose phosphorylated and split into 2 3-carbon compound. So that is the total funda. So let's see what happens. Initially you have glucose, and glucose is phosphorylated at the 6th carbon by the help of enzyme called as hexokinase, producing glucose 6-phosphate. And now this step is called as priming step, priming reaction. Why it is called priming reaction? See, you have other fuels also. So you consider other fuels, like for example diesel, petrol, or LPG or whatever. So if you want to burn and get energy out of them, initially you have to set fire to them, or you have to, in cars and all your bikes, you have to use ignition first. So a certain amount of energy is initially required to be spent on these fuels, so that later you could get a controlled set of energy. So same way here, you have to utilize ATP in the beginning. That's why it's called as priming reaction. So it is a priming. So we are priming the glucose to produce ATP later. So this is the first priming reaction, hexokinase reaction. Then what happens is glucose 6 is converted to fructose 6-phosphate by an isomerase enzyme. Then again, the next priming reaction happens, second priming reaction is where ATP is utilized and the enzyme is phosphofructokinase 1, and it forms fructose 1,6-bisphosphate. Okay, then we have the fourth enzyme, aldolase. What aldolase does is that now you have a 6-carbon compound, and in the 6th carbon there is a phosphate, and the first carbon there is a phosphate. Now it cleaves it equally. When I mean by equally, what I mean is that 3 carbons and a phosphate molecule goes to either side. So you can see 1 2 3 4 5 6th carbon with phosphate molecule, and 3 second and first carbon with the phosphate molecule goes to the next side. So it split it into two 3-carbon compounds. One is glyceraldehyde 3-phosphate and the other is dihydroxyacetone phosphate. Now this dihydroxyacetone phosphate is again converted back to glyceraldehyde 3-phosphate with the help of triose phosphate isomerase enzyme. So in the end of preparatory phase, what we have is two glyceraldehyde 3-phosphate molecules. Okay, so this fourth step is very important because this is where the actual splitting takes place. So it is this step which gives the name glycolysis. This entire process is named after this step, aldolase step, where you have the splitting. So glucose is split here. So in preparatory phase, two important things are happening. One is phosphorylation with the help of ATPs. So this phosphorylation increases the free energy content of the intermediates, and then splitting of 6-carbon molecule into two 3-carbon molecules. So this much has happened already, and now we are already in the preparatory phase. That means by definition glycolysis has completed here, but there is still more to glycolysis. We will see the next phase, that is the payoff phase. So now we have two glyceraldehyde 3-phosphate here. Okay, the main funda in this part of payoff phase is to somehow increase the potential energy or present in the carbon atoms of these compound. Okay, so that it is so high that that energy could be used to phosphorylate ADP. So that is a funda. So that energy is so high that it can phosphorylate ADP, producing ATP. So initially what happens is that we are oxidizing this with the help of a dehydrogenase enzyme, glyceraldehyde 3-phosphate dehydrogenase, and instead of a ATP molecule, now we are using an inorganic phosphate molecule. So we use an inorganic phosphate molecule, and now we have a phosphate bond created at first carbon as well, and this one first carbon as well. In the process, NAD plus is reduced to NADH, which is the reducing equivalent. And this reducing equivalent, if it runs through the electron transport chain, will produce ATP via a process called oxidative phosphorylation. So one NADH can produce either 1.5 or 2.5 ATP depending on how it reaches the mitochondria. So depending on which shuttle system it uses, it will either produce 1.5 ATP or it will produce 2.5 ATP. Since we have 2 NADH, so we will either get 3 ATP or 5 ATP. So now we have 1,3-bisphosphoglycerate. So there is phosphate bonds at both the end, and now we have a very high group transfer potential. Group transfer potential here. Which group transfer? Phosphate group transfer. Since we have very high group transfer potential, we can transfer our phosphate group to a compound having less group transfer potential, like our ADP. So it is transferred and ATP is formed. So this is the first ATP forming reaction, and it is a substrate level phosphorylation. So here I mentioned it is a oxidative phosphorylation because electron transport chain is involved. But here it is substrate level phosphorylation because there is no electron transport chain involved, there is no reducing equivalent involved. Rather, substrate with high group transfer potential donates the phosphate group to ADP, forming ATP. So that's why it's called substrate level phosphorylation. The enzyme involved is phosphoglycerate kinase. And now we have a 3-phosphoglycerate. So this is a 3-phosphoglycerate. Now this is a relatively less energy compound and it is a more stable compound. So we are going to shuffle the phosphate across and shuffle the bonds between these carbon, so that again we create a high energy or high potential bond. So let's see how that happens. So we have 3-phosphate glycerate, that is phosphate is in the third carbon. Now it is shifted to the second carbon. Okay, with the help of mutase enzyme. So we have a second carbon is phosphorylated now. So we have 2-phosphoglycerate. Then this 2-phosphoglycerate we remove a water molecule from them with the help of enolase enzyme, and we create a product called as phosphoenol pyruvate. So the water molecule between these two carbon molecules is removed out. Now we have sufficiently high group transfer potential, so that could be used to produce ATP with the help of pyruvate kinase, and the end product is pyruvate. So this is the end product of glycolysis. This is the second ATP forming reaction. This is the second substrate level phosphorylation. So here the funda was we raise the group transfer potential of the glyceraldehyde 3 phosphate so much so that we were able to produce ATP in two of the steps and NADH in the first step. So this is the payoff phase. So what are the clinical chemical transformations that were very important here? First is the degradation of carbon skeleton of glucose to yield pyruvate. That means splitting of carbon atom 6 carbon to 3 carbon. Next is phosphorylation of ADP to form ATP. From where did we, how we were able to phosphorylate ADP? With the help by creating high compounds with high phosphoryl group transfer potential. So we created compounds while splitting our um, 6 carbon molecule to 3 carbon molecule, we created compounds with high phosphoryl group transfer potential and used those compounds to phosphorylate ADP. Then in the process, we were also able to transfer hydride ions to NAD plus to form NADH. So these are the three major and more important chemical transformations of the glycolysis. Now here we can see a better overview. Here you have, see, glucose molecule, the first carbon is an aldehyde, there is a double bond carbon, and we have the last carbon here. And now we phosphorylated them. This is a part of priming step. So now we have phosphate at the sixth carbon. And now in order to split it into two equal parts, we require phosphate at the first carbon as well, but we have a double bond here, so it is not possible, it's not that easy to put a phosphate group here. So what did we do? We shifted the double bond to the second position, creating a fructose molecule. So now we have a fructose 6 phosphate. Now this first carbon is free, and now it could be phosphorylated. So we used ATP and we created a phosphate bond with the first carbon as well. So now we can easily split to give a 3 carbon molecule with phosphate, another 3 carbon molecule with phosphate, a symmetrical split. So we got a dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, and this could be again converted to glyceraldehyde 3 phosphate. In the end, we have 2 glyceraldehyde 3 phosphate. So that is the end of the preparatory phase. Next, what happens is that we are trying to improve the group transfer potential of this compound. So how did we do? We added another phosphate, this time with a not ATP but inorganic phosphate is added. At the same time, we also reduced NAD plus to NADH. So now we have a reducing equivalent also. This also can produce ATP when it is run through electron transport chain. Okay, so now we have a compound with two phosphates, and the first phosphate has very high group transfer potential, so that is utilized to produce ATP, and now we have produced relatively a stable compound. So in order to increase the potential energy between these bonds, what did we do? We rearranged the carbon and phosphate. So first the phosphate from third group is sent to the second group, then we do a dehydration reaction. So we remove the water molecule between these two carbons. So now the phosphate group has high phosphoryl group transfer potential, so it transfers it to ADP, and we have pyruvate in the end. So this is the entire funda. So the overall reactions could be written like this: glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate, yielding 2 pyruvate, 2 NADH, 2 H plus 2 ATP and 2 H2O. Here we know in preparatory phase we actually used 2 ATPs, but in the payoff phase we got 4 ATPs. So the net is 2 ATPs in the payoff phase. So that's why this reaction is like that. But only a little energy from this 6 carbon compound is trapped as NADH and ATP. Okay, majority of the energy still lies in the pyruvate. See, this 2 carbon compound, see 6 carbon compound is simply converted to 3 carbon compound, 3 carbon compounds. So unless it is converted to carbon dioxide and water, where the complete oxidation takes place, any fuel, if it has to be completely oxidized, it needs to be converted completely into carbon dioxide and water. So unless it is converted to carbon dioxide and water, the complete energy cannot be produced. So in glycolysis, the maximum energy that you can produce is 7 ATPs, and minimum is 2 ATP, and the maximum is 7 ATP. So the rest of the ATPs would be produced by further oxidation of pyruvate. So total, from a single glucose molecule, your body can produce about 30 to 32 ATPs. 30 to 32 ATPs can be produced from single glucose molecule. So though that energy still lies in the pyruvate molecule, and in the next step after glycolysis, further oxidation will occur to the pyruvate and it will produce the rest of the energy. So that's all about. Thank you. And if you have any suggestions, please put it in the comment. And thank you for your patient listening.